Welcome everyone to the next in the series of the Global Alliance Fireside Chats. I'm Kathryn North. I've been Vice Chair of the Global Alliance now for over 10 years. And I'm delighted today to be having a chat to Daniel MacArthur. Now, Daniel is well known to so many of you because of his groundbreaking work in the setup of Nomad. He's currently director of the Centre for Population Genomics in Australia, which is a joint initiative across two of our states, New South Wales and Victoria, led by the Garvan Institute and my own institute, the Murdoch Children's Research Institute. Daniel's work in Australia is very much focused on developing a more equitable approach to genomic medicine in Australia through diagnosis, prediction, and treatment of disease across all Australian communities, continuing the work that he's done internationally. Prior to coming back to Australia, Daniel spent 8 years in Boston, where he was co-director of the Medical and Population Genetics program at the Broad Institute of MIT and Harvard and Mass General Hospital. He co-directed the Broad Institute Centre for Mendelian Genomics with the chair of the Global Alliance, Heidi Rem, and that centre has sequenced and analysed genomic data from over 10,000 individuals from rare disease families and contributed to date to the discovery of more than 100 new rare disease genes. As I've mentioned, Daniel and his team led the development of the Genome Aggregation Database, or Nomad, which is the largest and most widely used data set of human exome and genome sequence data that we all use all the time. And before that, Daniel did his PhD with me. I've known Daniel since 2000, so before the Human Genome Project was completed. And he did his PhD with me in Sydney, which we'll talk a little bit about, and then a postdoc following that at the Wellcome Trust Sanger Institute in the UK, where he contributed to the analysis of the first ever population genome project, the 1000 Genome Project. So you can start to see a pattern from those early years of Daniel's very deep involvement in the development of our work across the human genome. So over to you for a few comments, Daniel.
Oh, thank you, Kathryn. Delighted to be part of this fireside chat. Although I was just saying earlier, it would be lovely to have an actual fire here because it is freezing in Sydney this morning. And thanks to everyone who's joined today. Before we get into the questions, I thought it might be useful to reciprocate by giving a bit of background on Kathryn for those who don't know her. Kathryn's career has focused on improving the diagnosis and the care of children with inherited neurological disorders, as well as physical and intellectual disabilities. For over a decade, Kathryn has been the director of the Murdoch Children's Research Institute in Melbourne. While she's been there, she played a critical leadership role in the establishment of genomic medicine in Australia, and as we'll discuss later, was really a key part in my decision to return to Australia back in 2019. Kathryn leads the Australian Genomics, which is a collaboration of over 100 institutes around the country that's focused on the implementation of genomics into clinical practice. And of course, she's also the Vice Chair of the Global Alliance of Genomics and Health, which is why we're here today. And she also co-leads, as part of that, the National Initiatives Forum, which is a program helping to coordinate genomics programs around the world, one country at a time. Kathryn's also responsible, as we'll discuss later, for teaching me pretty much everything I know about how to run a research group and collaborate as a researcher. So it's a little strange, but also really wonderful to be having this conversation today. I'm delighted to be part of it.
So Daniel, I know that you started your scientific focus with your Bachelor of Medical Science at the University of Sydney back in 1999, and we first met in the year 2000. So I was just reflecting with the Human Genome Project being completed in 2003 and thinking about the work you've done over the subsequent years. Your career really parallels the growth of human genomics as a discipline, and you've spent time in many of the leading genomics institutes around the world, and I've mentioned the Sanger and the Broad. So Could you start by telling us how you ended up working in genomics in the first place?
Yeah, it was we first met at the end of my first year at university. So this is in second year at university, actually. It was in 2000, where I applied for a summer studentship. I knew at the time that I was interested in figuring out how research worked. And so there was a notice board with a bunch of opportunities to do summer research programs. I applied for a few labs. You and I met. I remember being interviewed and deciding immediately this was the lab that I was going to work in. And so then spent 6 weeks over that summer working in the lab. And at that time, and this was pre-genome, the genome sequence being available, the work that I did over that summer was focused on studying a set of patients who had a set of muscular dystrophies, undiagnosed muscular dystrophies, where it was clear that they had been unwell. Many of them had been unwell for decades, but where we weren't able yet to have identified a genetic diagnosis. And over the course of that summer, to my surprise and amazement, I was able to go all the way from identifying for two of those patients that there was a particular protein missing based on staining their muscle, and then sequencing the gene and identifying a genetic diagnosis. And that ability to go for the first time for these patients from a muscle sample right the way through to a genetic diagnosis was thrilling. And the fact that we could do that before there was even a reference sequence in place was kind of amazing. But what that revealed to me was just how the degree to which being able to study DNA at scale was going to be able to change the lives of many patients. And so that was something that pulled me into this general space of genomics. And then during my PhD, honors year in PhD, I moved into a project in your lab focused on a really interesting gene called alpha-actinin 3, where we had this very strange genetic variant that results in the loss of function of that gene in over 1 billion humans worldwide. So a common polymorphism that creates a stop codon and knocks that gene out. And so over the course of my PhD, I was able to go from firstly collaborating with many others in the group in the Australian Institute of Sport, that particular genetic variation had an impact on human sporting performance, and was also able to make a knockout mouse and show that it had an impact on the function of the way that muscle worked in mice and in humans as well. And by the end of my PhD, I started to get really interested in this question of, given that we had this one common stop codon polymorphism in this one gene that had an impact on human biology, the question in my mind was how many more of those existed, and could we use what was then clearly the emerging power of genomics to explore that further. And so that's what pulled me in this direction of thinking about genome-wide analyses.
So I was just reflecting on your time as a summer student, and over that short period of a couple of months, finding those mutations in dysferlin at a time where we were doing Sanger sequencing one exon at a time. And I do remember when you gave your final talk as a summer student, I put up a slide of a needle in a haystack, and Daniel had been able to sort through so much information at that time to come up with the first genetic mutations we identified in dysferlin in my lab. And then the work on alpha-actinin 3, really over a number of years, and going from characterizing that polymorphism over multiple different populations, because the effects that we found around natural selection and its place in evolution was also quite interesting.
Yeah, that natural selection piece was cool, and that was a bit of an unexpected discovery. But by that point, we knew that this genetic change clearly affected human muscle biology, but it wasn't clear why it was that this loss of function mutation had become so common in the human population. And it seemed reasonable at the time to think that perhaps that had happened through some sort of process of natural selection, that there had been some kind of positive selection that had increased its frequency in different populations. But being able to, in those early days, actually bring together data from what was the first iteration then of the HapMap project, looking genome-wide at lots of different polymorphisms and seeing how they fit together, to actually study whether the pattern of polymorphism in the actinin 3 gene was different relative to the rest of the genome. That was what allowed us to then zoom in on the likelihood that this was actually under some form of positive selection. So that was definitely my first one of my first experiences of thinking genome scale, thinking about one gene first, but then looking at it in the context of the rest of the human genome.
I do remember one conversation we had during your PhD when you were deciding which path to go on, where your two interests were science and your love of science, but the other thing you were thinking about is whether you should become a scientific journalist. And I do remember giving you the advice at the time that either way, you really need to write lots of papers, and which I think has stood you in good stead. But I'd also note that Daniel got a Nature Genetics paper as one of the major outputs of his PhD before then taking up the position at the Sanger as a postdoc. But I also remember when we went to towards the end of your PhD, we went to the American Society of Human Genetics conference, and Francis Collins was one of the featured speakers talking about the completion of the Human Genome Project. And I just remember at the end of his talk, Daniel went up to have a chat to Francis Collins, and I think that was quite a pivotal conversation as well. So would you like to tell us about that?
Yeah, this is the first time I'd ever spoken to Francis. I had a few opportunities to speak with him later while I was in the US. But I was a scared little PhD student at that time, but I mustered the courage to walk up to Francis at the end of his talk, I think slightly to his surprise, and just asked him very quickly, "Francis, before you disappear, I'm thinking about what I would like to do for a postdoc project. Do you have any advice that you would give someone who was thinking about deciding on a project for their postdoctoral work?" And he paused only very briefly and then gave this very quick response, which was that what he suggested was finding a biological problem that was very interesting, and then discovering some way of applying these new next generation sequencing technologies to answer that problem at scale. And that general philosophical approach of being driven first by biological curiosity, but then making sure that you can find a way of aligning that question to the technology that at that time was very clear was going to be driving the next big wave of scientific discovery, was what ended up driving many of the decisions that I later made as part of my career. So yeah, Francis was not only had a huge impact, obviously, on the growth of human genomics in Australia, in the US more generally, and around the world, but had a massive influence on the way that I ended up making decisions around my career.
I'll never forget that talk with Francis saying the Human Genome Project will make it possible for researchers anywhere in the world, in any laboratories, to start making big discoveries, which has certainly been true. So then you moved to the Sanger and to work on the 1,000 Genome Project that led to your work at the Broad. So can you tell us a little bit more about your postdoc at the Sanger?
So I took Francis's advice to heart and decided at that time the thing I was interested in was this really intriguing discovery we'd made in the actinin 3 gene of a gene inactivating polymorphism that was common in the population. Clearly had an impact on human biology, but had somehow managed to spread around the world. And I was very interested in the question of how many more of these things might exist. So how many such gene inactivating or loss of function variants are actually found in the average human genome. And at the time, we had one human genome, just about. So this was back in around about 2007. There were a few more that were starting to be sequenced using next generation sequencing technologies. But the first population scale sequencing project, the 1,000 Genomes Project, was just underway. So at the time that I moved to the Sanger Institute, the opportunity was to start thinking about looking at these loss of function variants in the context of the first, at the time, 180 genomes that were being sequenced from three different populations of European, East Asian, and African ancestry. So I did my postdoc at the Sanger with Chris Tyler Smith, and I spent the 3.5 glorious years there in the beautiful countryside setting of the Sanger Institute down in Hingston, just south of Cambridge, looking at these first genome sequences with next generation sequencing technologies, which it must be said, because this was the very early days of Illumina sequencing and other technologies, were pretty rough. We had very high error rates. The community as a whole was really just figuring out how to make sense of the data that was emerging from the very short reads coming out of these new technologies. And I and the team had the daunting task of then trying to interpret the genetic changes we were finding, to map those onto genes, figure out which ones were actually going to disrupt the genes, and then filter through them one by one to identify all the different error modes and problems that had arisen that we had to get rid of in order to zoom in on the true loss of function variants that actually existed in the population. So that took quite a long time. It was really about 3.5 years of work. But at the end of that, we had an answer, which was that based on the technology we had at the time, that every human was walking around with at least 100 true loss of function polymorphisms that were actually reducing the function of the genes. The vast majority of those being rare, usually only found in the heterozygous state, but still that there were a surprising number of situations in all of our genomes where we were actually lacking the function of one or more genes. And that was the beginning of a really interesting question in my mind, which was then, could we use those to start exploring the function of human genes? Could we identify human knockouts who were lacking the expression of who were lacking the function of a particular gene, and then use them to understand the biology of that gene, and eventually potentially identify ways we could use that gene for therapeutics as well.
Mm.
And so that which was a great question, but was going to require much bigger data sets to tackle.
Yeah. So at that stage, I know there was a decision to make of what you did after your postdoc at Sanger. And Daniel had a fellowship from our Australian government that allows you to go overseas, but then also funds you to come back to Australia. But at the same time, Daniel was approached from the Broad Institute about taking a role there. And I remember that discussion that we had about that opportunity. And I do remember saying, even though I would have loved you to come back to Australia, I just said, take it, to be able to take the work and the ideas that you have to the scale that you can go to at the Broad Institute is such an amazing opportunity. So would you now like to talk about how what first lured you, apart from the obvious, to the Broad Institute, and how you set up and what your work there and the directions that you took?
Yeah, so the opportunity to move to the Broad came about through a conversation with an amazing guy, Mark Daly, who I first met at another American Society of Human Genetics meeting. He approached me and talked about a new unit he was building in Boston at Massachusetts General Hospital with an affiliation with the Broad Institute of MIT and Harvard, called the Analytic and Translational Genetics Unit. And I was sold pretty quickly. I had other opportunities to consider, of course, potential return to Australia. I'd also had conversations with some startup companies as well. But after talking through the options, it was very hard to imagine a place where I would be able to think about genomic questions at anything like the scale and with the type of intellectual rigor that Mark brought to these questions, other than the Broad. And so I was sold. My wife and I, and at the time our first child, then moved to Cambridge, Massachusetts, and that started my 8.5 years there at the Broad. And while I was there, there were a few different opportunities that came up.
What year was that?
So I moved in 2012, early 2012, and
Yeah.
this was, by that time, the Broad was already the largest genome sequencing production facility in the world. So it was an enormous amount of data that was being generated, but also had this incredible intellectual community that it still does, of course. So a real critical mass of people thinking deeply about how we could use all of this data rolling off these machines to answer interesting biological questions. And so as I settled into that role, there was a couple of different opportunities that came up. The first was I started to work with the Broad Genomics platform, led by Stacey Gabriel, to sequence at that time the exome, so just the protein coding bits of the genome for individuals affected by severe muscle diseases. And that was a bit of a chance opportunity. There was capacity available on Broad sequencing machines. And then as that became clear, I reached out to you at that time, Kathryn, and we were able to set up a collaboration to start to sequence the first of what became several hundred patients affected by undiagnosed muscle diseases, where we could take a look at their exome and try to figure out what the genetic cause was. And that was super fun because in fact, with the very first family that we sequenced, family A in that particular project, we were able to, thanks to the quality of the sequencing data that was being generated, and then this new analysis pipeline that we had built with Mark, to identify a brand new gene for muscle disease, that was then picked up and studied by an amazing clinician, Nigel Clark, and resulted in a fantastic collaborative publication. So I was like, oh man, this is easy. We can discover new genes as soon as we sequence any family. And the next few families were a little harder, but that was an incredibly productive collaboration.
Yeah. And I'm just reflecting on that time. I mean, you know, for me as an ex-PhD supervisor, it was really a case of the PhD students giving back, because my uncle Lek, who was also a PhD in my lab, came to join you at the Broad. But, you know, we'd accumulated by that time, I think 200 very well characterized families with severe neuromuscular disease, you know, including, you know, that family you'd originally worked on, the families you'd originally worked on back in your honours year. And just the productivity of having well phenotyped families with beautiful genotyping. It was just an incredibly exciting time in terms of gene discovery and finding diagnoses in families that, you know, I'd personally been following for over 15 years.
And that for me was a key lesson, and actually was the first time that I had explored what became a model that we ended up deploying on a much larger scale, which was the collaboration between our group bringing these kind of genomics and bioinformatics skills to bear, but then being able to work incredibly closely with teams that had that clinical domain expertise, so really knew the families, knew the underlying biology, could look at a gene and figure out how to make sense of that. That pairing, so long as you could build the right infrastructure, that pairing was the key to success in diagnosing those families. And so we had through that first collaboration, we built out a software platform called, at the time was called XBrowse, and later became what we now call Seeker, which was an online platform for analyzing data, where most importantly what it did was allow groups that didn't have a bioinformatics background, but had that clinical domain expertise, to be able to directly access the data and have a look at it, go through all the variants very easily and filter them in different ways, but then make their own decisions about which genetic changes were most likely to be causal. And that was the beginning of, you know, many collaborative relationships over the years with rare disease groups in Australia and around the world, following that same kind of model, that sort of deep collaborative model between the genomics and the rare disease work.
So we'll come to, you know, it's a good segue into talking about your development of the Centre for Mendelian Genomics and NOMAD. But I'm just going to put a personal reflection in here. I remember coming to visit you, I think it was in 2013 at the Broad. It was the year that I'd just taken over as director of the Murdoch Children's Research Institute, so I'd moved to Melbourne. But you introduced me at that time to one of your mentors, who was David Altshuler. And I remember spending, you know, an hour over lunch with David. And then he contacted me after that meeting and said, oh, we're setting up this thing called the Global Alliance for Genomics and Health. I'd love you to be involved. So that was actually, so I blame you, Daniel, for my now subsequent 10 years of involvement with the Global Alliance, initially leading what was the clinical working group for that. But it's just been a wonderful experience. But again, forging those links internationally has been, you know, really important from an Australian perspective in how we've set up genomics in Australia.
Yeah, I mean, guilty as charged. I definitely take responsibility for that introduction. But I will remember that meeting and how immediately afterwards David came to me and asked, "Do you think Kathryn would be interested in playing some role in this new Global Alliance thing we're putting together?" And I said, "Absolutely, you should talk to her and see if it's going to work." So yeah, I definitely take some responsibility there. But David was also a really critical figure for me as well. I was very fortunate actually in that first couple of years at the Broad to have both Mark and David providing mentorship, and really it was a conversation with David that triggered what became one of the most important projects to come out of my group at the Broad. At the time, we had started to sequence these rare disease patients from your team. And what was clear as we were looking through that data was that we needed much better data sets to compare those patient sequences against. The existing reference databases of human genetic variation were too small. The variant calling had not been done using best practice methods. So it could be really difficult to figure out for a given change found in a patient, whether it was actually a rare, potentially disease-causing variant or actually a common polymorphism in the population. And I knew that by that point, the Broad Institute had sequenced several tens of thousands of human exomes. And so I started to think about the opportunity of bringing all of that data together into some kind of publicly accessible reference database, but it wasn't 100% clear to me how that would work. And I'll always remember this half-hour conversation I had with David Altshuler where I laid out the opportunity and the challenge, and he really helped me think through in a very quick succession the model that would later become first the Exome Aggregation Consortium and later the Genome Aggregation Database, or GNOMAD, which involved collaborating with groups that were sequencing exomes and later genomes as part of common disease gene discovery projects, and working with them to bring all of that data together, thanks to the power of the analysis platforms at the Broad, into these big unified data sets where the summary data could then be publicly shared in a way that was ethically aligned with the consent that the participants had given, but that suddenly created this big reference data set that any clinician anywhere around the world could compare their patients against and figure out which variants were rare or common. And that realization, and this was David's genius, was his ability to figure out how to align incentives so that you could actually put together a big collaborative consortium where everyone ended up winning. The groups would allow their data to be reused in a way that was different from their original intent. And as a result, we were able to create a resource that could help any group anywhere in the world diagnose their patients, which was fantastic.
I remember those early years initially with the release of EXAC, because you and I would always tend up to catch up at least once or twice a year, either around American Society for Human Genetics or a visit to Australia. But I remember when your team was working, 24 hours a day to get that release, I think it was of EXAC, out to be presented at the American Society meeting. And I remember feeling very proud when you said you'd broken the cloud at the Broad Institute.
We certainly did some damage at that time. And it was a stressful period we had promised that we were going to announce the release of this new resource at the ASHG meeting. But it actually came down to literally the night before my presentation, sitting in a hotel room with a whole bunch of key people. Monka Lek from Australia had been an absolutely critical player in putting this data set together. Konrad Kaczewski and many others had also Konrad had helped to develop a brand new browser, a website that would deliver the first release of EXAC to anyone who wanted to access it. And so it came down to the very small hours of the morning just before my presentation, as we had our last chance at actually reloading the database to make it work. And amazingly, it worked. I was able to get up the next day and present the first release of EXAC to the community. People started to hit it immediately. So we had thousands of page views even that first day, and it survived. The website stayed up, somewhat to Conrad's surprise. And that was the beginning of what's now been many tens of millions of page views from groups all around the world using that data set to help to diagnose patients.
And tell us about the migration into EXAC becoming GNOMAD.
Yeah, so that first release was back in 2014, and then over the following couple of years, a few things happened. Firstly, the Broad sequenced a lot more exomes, so we're able to go from the 60,000 exomes that had been released in that first version of EXAC up to 128,000 when we're accessible a couple of years later. But also more whole genomes were being sequenced as well. So we started to think about a new version of EXAC that would include genome data as well. That meant we had to rename it because it was no longer the Exome Aggregation Consortium. And so hence the name, the Genome Aggregation Database, or GNOMAD, was born. And that first GNOMAD release in 2016 included 128,000 exomes and the first set of genomes, 15,000 human genomes as well. We were able to also work with Mike Tarkowski's team to bring together to do structural variant calling across those whole genomes too. So for the first time, we could also bring in other classes of variation into the analysis. And that was another crazy release, Nomad V2, another last minute scramble to get everything ready for announcement at ASHG, and again another amazing, remarkably successful launch. So yeah, that was and then that led to then Nomad V3, which was our first really big genome release with 75,000 human genomes that we launched back in 2019. And so the Nomad project continued to grow from strength to strength, and in fact still does. So although I'd left the project as lead in 2019 when I moved back to Australia, under Mark Daly and Heidi Ream's leadership and me and others as part of the steering committee, the project continues to produce and now has the most recent release has nearly 800,000 human exomes in it, which just blows my mind. It's such a big data set, and has had so much impact over the years on improving the diagnosis of patients.
It's fantastic. And you've mentioned Heidi. Tell us now about how your work in rare disease grew through the Centre of Mendelian Genomics.
So Heidi and I had started to work together on rare disease programs at the Broad. Actually, at the time, there wasn't any substantial coordinated rare disease program that had existed at the Broad Institute. A huge amount of work was being done in common complex disorders and cancer, but rare disease was still new. And so Heidi and I got together, and actually initially with some internal funding support from the Broad, started to build out a rare disease program, and then had this opportunity to put together a grant to propose to secure NIH funding at large scale, just over $13 million for a new centre, a centre for Mendelian Genomics that would be part of an existing consortium that had been running for the last 4 years. That grant was well received. Actually got a perfect score, which was fantastic.
Yeah, so I just note that perfect score on an NIH,
that's great.
which I think is remarkable.
We were very happy with that. In fact, initially we both thought it was a bit of a glitch in the system, but it was but no, that grant did great.
Have you both framed the report for that?
We have not, but we should. That's a good point.
It's just the sort of thing to keep in a drawer, and when you're having a down day, just bring that out and read it.
To look at every now and then. That's right. Yeah, that was a great result, and we were excited then to be part of this consortium. And that then allowed us to build up a real infrastructure and a fantastic team, including folks like Anne O'Donnell-Luria, a clinician scientist who had joined my group as a postdoc, but then played a really leading role in helping to build out this rare disease program at the Broad and make the Centre for Mendelian Genomics into a sort of a humming enterprise, working with groups all around the world who would send DNA samples from their families affected by undiagnosed genetic disease. We would then, working with the Broad platforms, generate typically exome, later some genome and RNA sequencing data as well. And then through the Seeker platform, then work directly with those clinical groups to find diagnoses. So in the time that I was there, that meant over 10,000 families that we were able to analyse, and more than 3,000 diagnoses that we were able to then return back to families. And of course, that work also now continues under leadership of Heidi, Anne, Mike Tarkowski, and others, continuing to deliver many thousands more diagnoses back to families. So that has meant through that combination of the Nomad project that allowed us to sort of look at human variation at a population scale, and then the fact that we had this engine that was working with groups around the world to look directly at the DNA of patients, we had an opportunity to bring those two data sets together and think about the various different ways in which we could use population scale data to tell us more about human variation and genes and how they worked, but then also to directly inform the way that we diagnosed and discovered new disease genes in patient data. So that was, it was an incredibly fun period of time at the Broad.
An amazing 8 years at the Broad. And then, as you said, in 2019, you headed back to Australia. So we'll talk about that now. But before we go into that, just to notice, we will have some time for questions, I think, although I think Daniel and I can talk forever. But please put any questions that you have in the Q&A. So Daniel, 2019, I remember we were having discussions around what your thinking was at the time. What was it that brought you back to Australia?
A lot of different factors. I mean, I had, although I have to say it was sad for me even to think about leaving the Broad. At that time, I had an amazing team, a whole bunch of people that I really loved working with, and collaborators there as well. But for multiple reasons, coming back to Australia in 2019 was becoming more and more appealing. Some of those reasons were personal. I had at that time, my wife and I had 3 kids. We were both keen to ensure our kids grew up with Australian accents and grew up back in Australia. So that was part of it. My
Do they
parents were
have
also
Australian accents now? Because they
they're
were,
getting there. The
yeah.
oldest I think has now almost lost his American accent, and the younger two are pretty Australian. So yeah, mission accomplished on that front. And my parents were also in their 70s, and I wanted to make sure we had a chance for my kids to grow up around them. But there was also big scientific changes and opportunities that were pulling me back as well. The growth of Nomad had been incredible, but one thing that had always haunted me a little as an Australian was that so many of the communities that live in Australia in large numbers were really not represented in the Nomad dataset. So these are groups like Australian Indigenous populations, groups from the Pacific, from Southeast Asia or the Middle East, for instance. And so that was a missing opportunity to help patients from those backgrounds. But there was also the fact that the whole landscape of genomics had shifted over the 12 years I've been away, and transformed in a way where it was now feasible to really think about national scale approaches to genomics, and that had changed to a large extent because of the work that you had led through the Australian Genomics program. So actually, I think it might be good to jump to you to give a little bit more context on how Australian Genomics had come together and what that did, because that was a key part of my decision to return.
Yeah, I mean, it was all around that time, 2013, 2014, as we were using genomics more and more for what we was in a research perspective. I knew that with my move to the Murdoch Children's Research Institute, which was initially established for genetics and then genomics research, that it wasn't really good enough that you could come to specialized centers, say, such as my clinic and my laboratory, and get a Rolls-Royce workup because of research. How do we start to think about taking genomics to scale and into clinical practice so that there is an equity of access to this, really transformative technology? And so we did some initial pilot studies across Victoria. We formed what was called the Melbourne Genomics Health Alliance at that stage, to demonstrate that, we looked at 100 kids with likely genetic diseases, and using standard of care at that time, getting around 10, 11% diagnostic rates. And then if you added in at that stage, it was exome sequencing in the same 100 kids. We lifted that diagnostic rate from 11% to 55%. And then looking at the health economics of that, the cost per diagnosis, even though genomics was expensive, we took the cost per diagnosis down from around 24,000 for a workup to $6,000 for a workup. So that also sparked the interest of government, because they could see that the implementation of this technology was really going to make a difference in terms of how we delivered healthcare. And so around the time when I was becoming very involved with the Global Alliance, there was an opportunity and a targeted call from our government to set up Australian Genomics, which was then, in terms of our setup, we're a federated nation with multiple states and also a single federal government. It's a complex healthcare system, but it was bringing together everyone around the country so that Australia could function as a single cohort of 27 million when you're looking at things like rare disease and cancer, and the need to build that data infrastructure and a commonality and national networks of people working together. So that's been the principle of Australian Genomics, which obviously is a driver project within Global Alliance. But that interplay from my perspective of us being part of the international collaborative community and being able to contribute our ideas of what needed to be done, but to, some of the standards that have been developed through Global Alliance, but then bringing international best practice back into Australia has just been, it allowed a major step forward. But back to you, Daniel. I think that what you've set up in Australia is a pretty unusual organisation because it spans across two major genomics institutes in Australia. So can you talk about the Centre for Population Genomics that you've established in terms of its setup, but also the amazing work that you're already doing?
Yeah, so I mean, I think the audience can see from that description of Australian genomics how important that was for my decision to return. And that, for me, that had been when I left Australia back in 2007. Genomics had really not been a thing in the Australian setting, and then by 2019, that had changed quite profoundly. But so the question was, in thinking about a return to Australia, what would the right model be? And I knew that this would require a different model from what my team had built at the Broad. But the thing that I was most keen on building was something that was truly national facing from the outset. That didn't feel as though it was focused on any one state or organization, but that had this sort of national facing and ideally global facing mindset from the beginning. And so as I started to have conversations with you and with others in Australia, my goal was to think about a model that was a partnership from the beginning. And that began a conversation that turned into the Centre for Population Genomics being built as a joint initiative between two great institutions, the Garvan Institute for Medical Research in Sydney and the Murdoch Children's Research Institute in Melbourne. At the time, the idea of having an organisation that was actually split across two cities that are pretty far apart, like an hour and a half flight between them, was pretty unusual. And it would require things like figuring out how to make a geographically distributed organisation work. And remembering these discussions were pre-COVID, the idea of having a highly remote first operating team was really quite
With
an
people
unusual.
working virtually, for heaven's sake, by Zoom.
Very strange idea. And so, but to the credit of both organisations, thanks to you and Chris Goodnow at the Garvan, who led the Garvan side of things, there was great enthusiasm about the idea, and so that resulted in the formation of this organisation. So we were able to kick that off in early 2020. I landed as my family and I landed in Sydney. There were bushfires burning, so the whole state of New South Wales was on fire. We had about a month to kind of settle in, a month of semi-normal life as my kids started school, and then this whole COVID thing happened, and that was disruptive in some ways, but actually in other ways really helped us and organisations in general think about how we could set up teams in a way that would be successful in that remote first environment. And that has been, I mean, in some ways, a weird kind of blessing for us is that it has actually helped us build that remote first infrastructure for the team as a whole. But the group, the Centre for Population Genomics, has now grown to nearly 40 individuals. We operate with people both in Sydney and Melbourne, and actually increasingly scattered across the Australian eastern seaboard, and a few employees in New Zealand as well. The major focus and the mission of the centre from the outset has been this idea of working with the broad Australian population to build a more equitable model for genomic medicine. And I see for that we have really focused on three major areas. The first of those is building a more representative genomic resource for Australian communities. We're calling this Our DNA, and this is a program that is currently recruiting individuals, particularly from Pacifica, Southeast Asian, and Middle Eastern backgrounds, as well as working with a really amazing national consortium of Indigenous genomics experts led by Alex Brown, to ensure that we also have good representation of Aboriginal and Torres Strait Islander communities as well within the resources that we build. So that work is ongoing, and at the end of that, we will have in phase one, a set of 10,000 genomes from underrepresented communities to help to fill some of those gaps that currently exist in Nomad and other resources, and also to contribute data back to federate data with the Nomad resource to ensure that data can be made available to the world and to patients around the world that would benefit from that for diagnosis. And so that's one huge area of focus. We also have a big...
I'll just I think what you're doing and what you're setting up there is so important because it's working within communities, but also getting a depth of specimen collection, so that you can do multi-omics eventually, and are consented to do that on these populations, which will be an incredible resource. And the second thing, you've mentioned Alex Brown and the Indigenous genomics. I mean, I agree that's been an amazing network that's of Indigenous-led genomics research. And your partnership with that group, I think, has been, really helpful to empower what is now an amazing group of Indigenous people that are generating some amazing information that will be of direct benefit to our Indigenous populations.
We're so fortunate to have Indigenous leaders working in this space in the Australian setting. Alex, for those who don't know him, he's an Indigenous man and an amazing clinician researcher. We also have other leaders like Azure Hermes as well, an Indigenous woman who plays a key role in driving the work of the National Centre for Indigenous Genomics in Canberra. And having leaders who really understand and work directly with these communities, who can then provide the vision for how genomics needs to work for those groups, where teams like mine that can help to fill in some of these gaps around genomics and informatics can then provide support. I think that's the only way for Indigenous genomics to really get up and off the ground. And I still feel lucky that we've been able to have leaders like Alex and Azure to drive that work.
Yeah, no, it's a great model, and it's come so far over the last 5 years. So
Absolutely.
tell us, and again, if people would like to ask questions, now is the time to put them in Q&A. But I've still got a lot of questions for you, Daniel. Tell us also about the other work that's exciting you in the Centre for Population Genomics, because our DNA it's a big arm, looking at bringing equity into genomics, and that idea of how there will end up being, multiple cohorts around the world that can grow. We're starting to talk about a network of nomads to improve the reference databases. But tell us about some of your other work, particularly in rare disease.
Yeah, absolutely. So we have a big program in the rare disease space now, which continues a lot of the work that my team was doing at the Broad, but built in with this wonderful network of Australian rare disease researchers. It's led by Cass Simons, who was previously a faculty member at the Murdoch Children's, who's really built up now within the centre a program focused on rare disease that now has more than 5,000 individuals from undiagnosed rare disease families from around Australia, all sequenced mostly with genomes, some exomes as well, but all processed in the same way, and all of that data in a single unified resource that allows us to think about running joint analyses and new diagnostic methods across that entire data set. And one thing that Cass's team has built, particularly in collaboration with Zanitza Stark's group at the Victorian Clinical Genetics Service, and in a collaboration with the Broad Institute and Microsoft Research, is an automated approach to diagnosis that really helps to automate some of those key early steps in making sense of the variants that we see in patient genomes, and identify the low-hanging fruit as quickly and accurately as possible, so that we can not just find results faster, but most importantly, so that we can go through this process of reanalysis, so that if we don't find an answer in our first pass through a patient's genome, rather than that data being left on the shelf and not looked at in some cases for years, we can make sure that there's a system that's running in the background, constantly checking against the biggest updates, the latest updates on variant and gene discoveries, or new methods for variant calling, and integrating those so that we can, as soon as a new variant is discovered that could explain that patient's condition, that can be flagged and given to the clinical lab to allow that to be confirmed and then returned. And that has really been, it's not just impactful in its own right, so it's already made more than 150 diagnoses for Australian patients, but it's also now the foundation for work that that Cass's team will be leading, working with Microsoft and others around integrating new AI tools into that process of automated reanalysis. Something I'm personally super excited about, because I think this is one space where this very rapidly moving set of new technologies we can see in the AI space can be brought into clinical practice very quickly and effectively, and that is not only enabled by the speed of technology and the scale of these data sets, but also the fact that in Australia we do have these super productive partnerships with clinical labs, including particularly the Victorian Clinical Genetics Service, so that we can take new things that we're building and put them into practice with clinical patients as quickly as possible.
Give us some Can you give us an example from that reanalysis perspective of how that pipeline works, just with a real patient?
Absolutely. So it's a good example. We've now got a number of cases like this where there's a patient who was undiagnosed the first time round. We have built a pipeline that basically just goes through and quickly checks using the family structure and their genetic data for things like known disease mutations in known genes or loss of function mutations in known genes, and flags those very quickly for a return. A great example that Cass likes to talk about a lot is a family where it was a couple of years since their initial diagnostic scan. Nothing was found in that first pass through the genome, even though a very talented curation team at VCGS went through that data. But then 2 years later, with the first pass of our automated reanalysis project, again, nothing. But then just a month after that, thanks to the discovery of a new disease gene that was published in the intervening period, when we next ran the pipeline, that then identified a clearly causal genetic change in that newly discovered disease gene that we were then able to confirm and rapidly return back to the family. And these are the types of stories I think that we miss far too often, where basically in that case we were able to pick that up almost immediately within weeks of that gene being discovered and reported. But of course, for the vast majority of patients around the world, that regular reanalysis process is not a feature of the way that we analyze data, for both for financial reasons and for technical reasons. And so for many of those patients, they won't get an answer. Instead, their data sits there in archival storage. And
or
it's not looked at again.
The doctor asks again in 2 years' time when they come back. Come back. Is there anything new? Yeah,
precisely. And so, given how long these families have been waiting for answers and how much of a difference it can really make to get a diagnosis, I think we owe it to families to be returning these answers as quickly as we can. And I think this automated approach to reanalysis and increasingly making sure that we can integrate new AI tools to make that as accurate and as safe as possible will make sure that we can get answers back to those families as fast as possible.
And so how frequently does that reanalysis occur?
So in theory, you can do it
in real time, theoretically?
Yeah. You can do it as often as you want. It's remarkably cheap and quick to run once the system's been set up. Our view is that really we need to move towards a model of what Cass would call continuous reanalysis, which basically means that as soon as a new data becomes available, for instance, if there's a new release of the ClinVar resource or a new disease gene has been discovered, that should then trigger a reanalysis of all of the data that sits within a particular lab's resource. And that means that basically the moment that data is available that could lead to a diagnosis, we have a tentative answer that can be flagged and looked at by a curator. And we've tried to tune the system in a way that means that it doesn't generate a lot of noise. So it really finds the drop-dead obvious signals, but is not giving a whole bunch of false results that labs need to chase up. It's just really a very clean approach to finding very likely causal variants. So that's the start of what I think will be a really exciting process of increasingly not displacing human expertise in this process, but automating and synthesizing evidence in a way that makes it as easy as possible and as cheap as possible to get answers back into the hands of families as fast as possible.
Quickly as possible. It's come a long way since your first foray in this area back in 2000, Sanger sequencing a series of patients. There's a question from Monica. How long do you think it'll be until the automated reanalysis becomes standard practice?
We're in the process of packaging up the software that we've built in a way that would make it easy for other groups to deploy. I would hope, given the impact of this type of approach, this automated reanalysis approach, that it will pretty rapidly become the case that that most labs around the world will want to get this in practice as quickly as they can. And I know certainly in the Australian setting, we've got a number of labs that will be deploying it as part of their standard practice very rapidly, from VCGS to South Australia as a starting point, and then beyond. My sense is that the biggest obstacle here will be reimbursement and making sure that we actually have the right pay structures in place, and this will vary obviously by jurisdiction and things like insurance practices, to ensure that there actually are the right incentives in place for patients to be able to get an answer quickly. So the over the course of the next couple of years, the barriers will not be technical. They will be the standard operational and financial challenges that face any healthcare implementation process.
And just as a plug, that's something that we're looking at in our National Initiatives Forum, is how we can feed this data, the health economic data, to inform health technology assessment in different countries, so that this more rapidly, there is the evidence there to start to drive that into being adopted into clinical practice as standard of care. So last minute, Daniel, what's most exciting you about the future?
So I think from my perspective, we are living through what is unquestionably the most exciting period in the history of medicine, I would argue, and that's because of this convergence of amazing change in progress. That's right. I mean, we're biased, of course, but we have this incredible change in genomic technologies, in data science that allows us to make sense of that data, and then in therapeutics, and with the advent of new sort of genome-focused therapeutic strategies. For me, I see, diagnosis of rare disease families is absolutely critical. It's the first step in that process towards getting more and more of those families into a situation where they can actually get access to the therapies they need to help their kids. But I think that combined speed of change in our ability to diagnose, and then our ability to generate and get approved targeted therapeutics that can actually help these children, is the most exciting thing. And so over the next decade, I'm looking forward to seeing a process where we can get every family that needs a diagnosis a specific genetic answer, and then for as many of those families as possible, actually get the right therapies in place, so that we can actually address the specific molecular defects in ways that provide material help and support to those families. The next decade is going to be very, very exciting indeed.
Indeed, bigger and better. Daniel, thank you so much. It's been, as always, a pleasure to chat against our virtual imaginary fire. But I think just amazing to reflect on, now you're 23 years in the business of genomics and the amazing contributions that you've made. But I agree with you. I think the best is yet to come, and that really is being able to drive this genomics revolution into healthcare is very, very exciting, and certainly from the Global Alliance perspective, I think that focusing on that delivery into healthcare is just so important as well. So thank you.
Well, thank you both for a great discussion today, but also, of course, for everything you've done to teach me and to help me on this journey. It's been a fun conversation, but also a pretty fun ride over the last couple of decades together.
Absolutely. So thank you, everyone, for joining our fireside chat. It will also be available as a video. I'm sure the Global Alliance will circulate the link as well. But thank you, Daniel, and I'll see you soon.
Thanks, everyone. See you guys.
Bye.